And it's my pleasure to introduce Dr. Bernard Meunier, who is the Director of Research at the French National Center for Scientific Research, CNRS, in Toulouse, France. And he has a very long and prolific career and recipient of many honors and awards, among which are the membership of the French Academy of Sciences and a Knight of the Legion of Honor. The focus of his current research is developing copper chelators as therapeutics for Alzheimer's disease. And the podium is all yours. Nice meeting you.
Okay, thank you for the introduction, and thank you to Rams for the invitation. It's a real pleasure to be part of this very interesting symposium. So let's start with a molecule that nickname of this molecule is TDMQ. They are very specific for copper chelation. We consider that this molecule can be a reasonable drug candidate for Alzheimer's disease. As you know, creating new drugs for Alzheimer to cure Alzheimer's disease is a very, very challenging domain. As you know, for the last 20 years, more or less, 100% of all attempts failed. So it's very depressing, more or less. The last approved anti-malarial drug was the one which has been named memantine in early 2003. Chinese FDA approved oligosaccharide in 2 years ago, but up to now, I've not been able to check if this molecule has been used outside of China. We don't have any full details about the clinical trials and activity of the molecule. It's supposed to interact with the intestines and the microbiota, and then since the microbiota is the second brain, it should go to the first brain. Very complicated. Very recently, FDA approved an anti-monoclonal antibody from Biogen in June. That was very controversial because apparently there is no real improvement about cognition, and there is some reduction of the amyloid plaques, but many MDs involved in clinical trials with Alzheimer are questioning this drug. So the starting point of my research is based on this slide. It's an old slide, but you know that there is a copper accumulation in plaques after post-mortem analysis of brain. Person will get the Alzheimer's disease. There is also an accumulation in the red labels for iron and for zinc. Compared to the normal situation, it's not a small modification. That means apparently the amyloid plaques are able to accumulate a huge amount of copper, iron, and zinc. And the problem with copper and zinc, and especially with copper, these two redox active metal ions are easily reduced by any endogenous reducing agent. That means you can start redox chemistry, and as described, reported already by Astrid, you can generate reactive oxygen species. As you know, this is a classical scheme for the generation of reactive oxygen species. You go down to hydroxy radicals. There is no way to escape from damages created by hydroxyl radicals. You can control hydrogen peroxide with catalysis and so on, but for hydroxyl radical H dot, there is no way to. So we can consider that hydroxyl radical may be at the origin of the irreversible cell damages and probably to the death of neurons. Okay, so the working hypothesis is as follows. Aging, from aging, you get post-transcription and post-reduction modification, for example, glycation of proteins or enzyme, rupture of homeostasis of copper. That means you get accumulation of copper on amyloid. then you get the RAS production, neuron damages, microglia increase, inflammation response. Then it's a long process, but at the end, this is probably the way to describe, one of the ways to describe the evolution of Alzheimer's disease. So having that in mind, we decided with other people, we are not the only one to do that. And Ashley Bush was also involved in that game, and other people, nearly for the last 15 years, to design specific copper chelator. We started in 2004 with a series of tetradentate ligand based on bis-8 aminoquinolines, which are very specific for copper, but very small chelation with zinc and iron. Okay, so we get a patent, we get everything on that, and we try to prove that this molecule were active on mouse model. We did at the beginning experiment with transgenic mice, but transgenic mice are, in my opinion, not the best model for to, let's say, select a drug candidate for Alzheimer's disease. It's a complicated disease, and we have to remember that Alzheimer's is not a genetic disease. 98% are sporadic, so it's a non-genetic disease. It's a disease which is related to aging, which is a complicated process. So when you create a transgenic model, you introduce hypotheses about genetics. And how many genes do we need to modify in the mice to generate a good model? This is not easy to understand. So we start with the idea of injecting amyloids in the brain of normal mice, just ICV injection. And the ICV injection, okay, just with 142 monomers or oligomers. And we created a loss of episodic memory. The interesting point, if you do the same thing with the reverse peptide, the 42-1, there is no loss of episodic memory. This is a good control experiment to prove that when you inject in the normal brain an excess of amyloid, you create a problem. With one of our drugs, which is in red, we were able to restore on this ICV model the episodic memory after a few weeks, and we gave the molecule by oral gavage. So it's an oral route administration, which is interesting for a future drug. And the quantity was about 12 milligrams per kilo, 9 doses over a period of 20 days, and we were able to reverse the loss of episodic memory. So we were happy for a while with this molecule, but when we start to think about the development of this bis aminoquinoline, the solubility was not good enough. Molecules were able to cross the BBB, but so we decided to simplify the molecule, and we created, in collaboration with people in my group in Guangzhou in China. I'm working as a retired CNRS fellow in China as guest professor, and we created this new type of ligands based on a single aminoquinoline entity with a side chain in order to get a good, efficient tetradentate chelator. The key point in order to get copper selectivity is to have 4 nitrogen atoms and an aminoquinoline, not an hydroxyquinoline. Hydroxyquinoline provides you non-specific ligands. So we check more or less everything on this molecule, and we published that in, it's interesting to see the huge difference of affinity between copper and zinc. If you look to the red arrows, for TDMQ20, you have 12 order of magnitude of difference between copper and zinc chelation. 12 order of magnitude. It's a huge difference. If you go to the same molecule with an hydroxyquinoline, you have a medium and average chelation, which is half of the value which are here. So then we decided to look at the X-ray structure to be sure that we have a tetradentate ligand, which is the case, no problem. And then we were interested to look about the possibility to inhibit the production of reactive oxygen species when you have copper chelated by amyloids in the presence of a reducing agent. So if you have amyloid copper and a reducing agent, you generate hydrogen peroxide and you generate hydroxy radicals. So the way to monitor the conformation of this reactive oxygen species is to monitor the decrease of the ascorbate, the reducing agent, for example. And when you add, after, let's say, less than 10 minutes, 8 minutes, some TDMQ20, the tetradentate ligand specific for copper, you see that you stop the production, stop the consumption of ascorbate. And you can do that even in the large excess of zinc. Even with a large excess of zinc, it's still possible to inhibit the production of reactive oxygen species. This is, let's say, chemical studies in a chemical lab. So what are the pharmacological activity of this molecule? We try some experiments still on the bench of a chemistry lab to be able to demonstrate that these ligands are able to extract copper from amyloids, and then the copper linked to the ligand can be reduced with glutathione and liberated the copper to create a complex with glutathione. So it's interesting to have a ligand which is able to act in a small amount as a ligand able to extract copper from amyloids and to recycle copper in the normal situation. The idea is not to extract copper from the brain. The idea is to have a molecule which is able to regulate the copper and to avoid the accumulation in the amyloid. Now, can we look about the memory modification with these ligands? So we decided to check the pharmacological activity of this TDMQ, and especially the TDMQ20 that we selected, on 3 different mouse models. We published that at the beginning of this year in the ACS Chemical Neuroscience. And we have 2 non-transgenic mice models. The ICV model, where we had the injection of copper on the A-beta 142, sorry, just one single injection in both lateral ventricles. Then we have the so-called HIPPO copper amyloid models, where we have a single injection of copper amyloid 142 in the hippocampus. So 2 non-transgenic models. We treated the mice with 8 doses at 10 milligrams per kilo by oral administration from day 4 to day 20. So at the end of the 3rd week, we checked the activity of the molecule with experiment on how to monitor the memory inhibition or not. And then we compare the data with regular transgenic mice model, the one which is used by more or less everybody, where we had 3 months old transgenic mice that we treated for 2 months, from 3 to 5. And then we checked the memory troubles with novel object recognition, Y-maze assay, open field assay, elevated plus maze, and so on. We published that, And I want just to share with you today a few slides about novel object recognition. You can see on the ICV model that the untreated mice, okay, which is the second column, you have a loss of the recognition index. And then when you have the treatment with TDMQ, you can see that we restore, we are able to, in fact, inhibit the loss of memory problems. And the CQ is a control with cleokinol, a molecule which has been used by Ashley Bush nearly 15 years ago. So you can see that for the ICV model, we have good data. With the hypo model, we are also good data. And if you compare now with the data obtained with the transgenic mice, you can see that from the data obtained with the transgenic mice, it's very difficult to see real differences. In fact, there are no differences. So the transgenic mice models are not suitable models for doing pharmacology. Maybe to study the disease, maybe. But to select a molecule, you need a model, animal model, where you are able to prove that there is a modification on the memory. Otherwise, there is no way to select a molecule if you don't have a good animal models. Let's see the open field assay. It's the same again. TDMQ20 is able to inhibit the troubles of memory troubles. And with the transgenic mice model, we have some data, okay. But all in all, with the different assays that we did, or different experiments that we did, I feel that the non-transgenic mice model are suitable models to get, let's say, a good selection of drug candidates. So, are we able to reduce the production of malonaldehyde, which is the signature of oxidative damage in the brain? And the answer is yes. With the ICV model, you can see that we reduce the production of malonaldehyde to the level of the control animals. It's also more or less the same with the transgenic mice model. So what we have been able to prove, we have been able to prove that these chelating agents are able to reduce the memory troubles, able to reduce the signature of the reduction of oxidative damages. Okay. So here we are. Now we have some summary of the, let's say, the different quality of this molecule. Let's go to chemical synthesis is relatively easy and robust. These are molecules which are EMS negative. The cardio safety is reasonable. The metabolism by human microsome or human hepatocytes is about 50% within 2 hours. CACO data indicates that they are able to cross a membrane. And we are now continue to work on that. We would like to have a biomarker that can be used for clinical trials if this molecule can be developed by a pharmaceutical company. I created a biotech company 20 years ago when I was younger, and with my age, I have no time to create a new biotech, but and I'll be sure that there will be people able to do that and to develop this molecule. We deeply need new drug candidate for Alzheimer's disease, which will be able to inhibit the progression of the disease in the early stage of the Alzheimer's disease. With the non-transgenic mice model that we created, I feel that these models are also good models to mimic the early stage of the disease. A good drug is a drug which is able to stop the evolution of the disease, because after a few years of evolution of the disease, there is no chance to get a medical solution. These are the name of the people and the place where the work has been performed. I have a very nice collaboration with my group in Guangzhou, in the University of Technology of Guangdong, and also a good collaboration with people of the Shenzhen University, Professor also Guillaume Liu, and I'm really enjoy my collaboration with my former collaborators, Anne Robert and Yan Liu, and many thanks for your attention. Ready to answer any questions. Thank you. I think I'm on time.
Thank you very much. And thank you for being so much on time. That's really very nice. So we can enjoy answering questions. And the first one is from a Christian. He says that I'm a little surprised that 5 times FAD mouse model showed so little effect in behavior. How long did you wait before measuring behavior?
That's many questions about transgenic mice, as you can imagine. We use 3-month-old transgenic mice model, and we treated these mice from month 3 to month 5 for 2 months.
Another question which you have is from anonymous attendee. Does this chelator bind to calcium?
I'm not sure. Can you repeat the question? I'm not sure to understand the question.
It asks whether
I heard that there is somebody trying to make
a I'm sorry. Does the chelator bind to calcium?
I'm sorry.
Does your chelator binds to the calcium?
Oh, hello? Binding to the calcium.
Yeah. Does it bind to the calcium?
Oh, wait, no. There is this nitrogen ligand, a very poor ligand for calcium, sodium, whatever it is, or potassium. We publish an article where we have been able to demonstrate, for example, that these ligands are not able to extract cobalt from cobalamin, vitamin B12, and so on. And so these ligands are very specific for copper ligand. If you want to bind calcium, you need, for example, anionic ligands like EDTA or things like that. Molybdenum, tetratiomolybdate is able to bind calcium and so on, but not this kind of neutral ligands.
Thank you so much. You got another question. It was in the Q&A section from Ashwin Ambi. A great talk. Does the TMQ lower amyloid content as well? And does it chelate copper from other important copper binding enzymes in the brain? So there are 2 questions. It's about, yeah.
That's an important question. We have a huge amount of copper enzyme, tyrosinase and so on, which are involved in the production of neuromediators. But we know that nature is well organized. The affinity constant of copper, when copper is incorporated in this metalloenzyme, is very high. So it's not possible to remove with such ligand, even with a very high affinity constant for copper from metalloenzyme or chaperone protein, which are involved as a copper carrier proteins. So there is a gap, a huge gap between chelation extraction of copper from amyloid, which is a weak binding system, compared to copper, which is incorporated in metalloenzyme. And if you inject a huge amount of TDMQ20 on a normal mice to check the toxicity, we don't see any toxicity at, let's say, concentration around 300 milligram per kilo, compared to the pharmacology dose, which is 10 milligram per kilo. So we have no toxic effect at high concentrations. That means we are not perturbating any fundamental enzymatic system.
And you have another very interesting question from Livio Mirica, great talk. Can you please speculate a bit more on why does your compound bind so much more strongly to copper versus zinc?
Oh, from the coordination point of view, I have to say there is a strong difference between the same ligand with an amino group on the 8th position of the quinoline compared to an hydroxy. When you have an hydroxy, you have non-selectivity for copper and zinc, and the affinity constant are more or less the same. When you don't, you replace this hydroxy group for an amino group, then you get the selectivity. Now, how to understand? Maybe I have to get a collaboration with a good theoretician. A good theoretician may be able to explain that. I have experimental facts, but I don't have a real good theoretical explanation in terms of TDFQ calculation and so on. That should be done. Maybe with Sesson Shaikh in Jerusalem, which is one of my good colleagues, expert about calculation on the metal-oxo species. We published a nice paper 10 years ago. I have to get Sesson Shaikh involved in that. But it's an open question. Very good question.
Yeah, thank you. So there is a question from anonymous attendee. Can you please comment on the role of the oxidation state of copper in fibrillation of amyloid? So which of the oxidation states you think it's important for amyloid?
When you have when you add copper-2 to amyloid, okay, you have a stable system. Then you start to add ascorbate, and you reduce the copper-2 to copper-1, and then copper-1 provide an electron to molecular oxygen, and you do that in a catalytic fashion. Okay? So then when you extract the copper-2 from the amyloid with TDMQ, you get a copper-2 TDMQ complex, which is stable. Unless you add a good reducing agent, able to destabilize this copper-2 TDMQ complex by reduction. Glutathione is good for that. Ascorbate is not as good, but glutathione is good for that. Then you are able to remove the copper from TDMQ and to get a copper glutathione complex, where we publish a paper on that.
Yeah, thank you so much for really great talk, and thanks. I thank the audience for the exciting questions. We went through most of them, and to keep up with the time, I would like to move on to our next speaker.